Welcome back. Let's continue to learn the placenta. Structure of placenta. The placenta is a disc-shaped structure composed of villus chorion of fetus and the decidual bacillus of the mother. The placenta of a full-term fetus is about 500 grams in weight, 15 to 20 centimeters in diameter, thick in the center and thin at the periphery, with an average thickness of about 2.5 centimeters. The fetal surface of placenta is smooth and covered with amnion. The umbilical cord is attached to the center or slightly slanted. Through amnion, radial branches of umbilical blood vessels can be seen. The maternal surface of the placenta is rough, which is the exfoliated decidual bacillus. There are 15 to 30 placenta lobules, separated by shallow sulcus. The placenta has two components, villus chorion and decidual bacillus. In the vertical section of placenta, the conjunctive tissue of chorion is seen on the amnion, and the branches of umbilical blood vessels run along them. There are 40 to 60 villi in the chorionic plate and many small villi in the stem villus. In this picture, you can see that the end of stem villus is fixed to the decidual by cytotrophoblast shell. The branches of umbilicus vessels enter the villus along the stem villus and form capillaries. There are villus spaces between the stem villi. The short septum composed of decidual bacillus extends into it, which is called placental septum. Placental septum divides the placenta into 15 to 30 placental lobules, each of which contains 1 to 2 stem villi and branches. The branches of uterine spiral artery and uterine vein open in the villus space, so the villus space is full of maternal blood and the villus is soaked in it. There are two blood circulation systems in placenta, maternal and fetal. The maternal arterial blood flows into the villus space from the uterine spiral artery. After material exchange with the fetal blood in the villus, it flows back to the mother through the uterine vein. The blood of fetus flows into villus capillaries through umbilical artery and its branches, and exchanges substances with the maternal blood in the villus space, so it becomes arterial blood and then flows back to the fetus through umbilical vein. The blood of the mother and fetus circulate in their own closed channels, which are not mixed with each other, but can exchange substances. The circulation of the fetus and the mother are separated by the placental membrane, consisting of fetal tissues. Fetal placental circulation. The oxygenated blood leaves the fetus and passes through the umbilical arteries to the placenta, where the cord attaches to the placenta. The blood vessels form an extensive arterial capillary venous system within the villus. The oxygenated fetal blood passes into thin wall veins that flow the placental arteries back to the site of attachment of the umbilical cord, where they converge to form the umbilical vein. This large vessel carries the oxygenated blood to the fetus. Maternal placental circulation. The blood in the intervillus space is temporarily outside the maternal circulation system. It enters the intervillus space through 80 to 100 spiral arteries. The maternal blood eventually enters the endometrial veins. The structure through which fetal blood and maternal blood exchange substances in the placenta, called placenta barrier or placenta membrane. The early placental barrier consists of syncytial trophoblast, cytotrophoblast, and basement membrane, thin layer of chorionic connective tissue, capillary, basement membrane, and endothelium. This animation displays that the process of placental membrane thinning. You can see the changing of the red circle, which is the placental barrier, in later stage of development, because the cytotrophoblast disappears in some parts, and the syncytial trophoblast is only a thin layer of cytoplasm in some parts. The placental barrier becomes very thin, and only the chorionic capillary endothelium, the thin layer of syncytial trophoblast, and the basement membrane are separated between the fetal blood and the maternal blood, which is more conducive to material exchange. Functions of placenta include two aspects: material exchange and endocrine secretion. And all these activities are essential for maintaining pregnancy and promoting normal embryonic and fetal development. Material exchange or placental transfer. Almost all materials are transported across the placental membrane by one of the following four mechanisms: simple diffusion, facilitated diffusion, active transport, panocytosis. The transported materials include gases, nutrients, hormones, electrolytes, antibodies, waste products, drugs, infectious agents, and so on. The fetus obtains nutrients and oxygen from the mother blood through the placenta and discharge metabolites and carbon dioxide. Therefore, placenta has the function of small intestine, lung, and kidney. Immunoglobulin G in maternal blood can enter the fetus through placental membrane so that the fetus and newborn have a certain degree of immunity. Because some drugs, viruses, and hormones can pass through the placental membrane and affect the development of fetus, pregnant women need to use drugs carefully and prevent infection. Endocrine function. The syncytial trophoblast of placenta can secrete several hormones, which plays an important role in maintaining pregnancy. The hormones are as follows: human chorionic gonadotropin, HCG, which is similar to luteinizing hormone, can promote the growth and development of maternal corpus luteum to maintain pregnancy. HCG began to secrete in the second week of pregnancy, peaked in the 8th week, and then decreased gradually. HCG can be detected in urine of pregnant women, so it can be used to detect early pregnancy. Human placental prolactin can not only promote the growth and development of mammary gland, but also promote the growth and development of fetus. Human placental prolactin began to secrete in the 2nd month of pregnancy and reached its peak in the 8th month until delivery. Steroid hormones, progesterone and estrogen, began to secrete in the 4th month of pregnancy and then gradually increased. After corpus luteum regression in the mother's ovary, these two hormones of placenta continue to maintain pregnancy. High levels of estrogen and progesterone have immunosuppressive effects, which are believed to be an important reason why the maternal immune system does not reject embryos with antigenicity.